Welcome everyone to El Zoominario. Today we're having Helena Jaramillo Mesa. Helena is from Medellin, Colombia, and for her undergrad, she studied biomedical engineering at Universidad Nacional de Colombia in Medellin, and she has a master's degree in biotechnology from the same university, in which she worked in the characterization of viruses infecting tropical fruit crops. After that, she came here to UW-Madison to pursue a PhD in plant pathology, working also with viruses, but focusing on the molecular mechanisms of the protein production. So thank you very much, Elena, and we're really excited to have you here. So please.
Thank you so much for having me. Like Claudia mentioned, I am a PhD student candidate here at UW-Madison in the Department of Plant Pathology, and I'm here to talk to you a little bit about plant viruses and how they produce their proteins. So let's start with what is a virus. So viruses are obligate intracellular genetic parasites that can only replicate inside a living cell and that contain their genetic material in a viral particle. Viruses can move on their own. They cannot generate energy or synthesize their own proteins. So they basically are absolutely dependent on a cell for everything to complete their replication cycle. You can see here in scale a viral particle, an example of a viral particle in the context of a cell. So viruses are very small particles. They range from 30 nanometers to 200 nanometers in diameter. So that's like 1/10 the size of a bacterium. And just to give you some context so that you can see it in your head, 1 centimeter would be the size of a wasp, and 1 millimeter would be the size of a small ant. And up until around here is how far we can see with the human eye. So beyond this point, we need another technique like a microscopy to be able to observe different things. Viruses, the vast majority of viruses cannot be seen even under light microscopy because they are so small. So we need to find alternative ways like electron microscopy to be able to observe them. Viruses are very diverse in their sizes. So we have very small viruses like parvoviruses that cause diseases in dogs. And we have 50 times that in viruses like Ebola virus that causes hemorrhagic fevers. But even within this, we all the time find new viruses that keep that makes us rethink and redefine what viruses are. But although they're very diverse, all viruses have common properties. And so I'm going to talk to you a little bit about what makes a virus. So first of all, they have a genetic material that could be either DNA or RNA. And that contains their genome. And in their genome, they have all the things that they need in order to produce the proteins or the molecules they need to complete their life cycles. That genome can be also very variable in size. It ranges from around 4,000 nucleotides to 1,200,000 nucleotides. And again, to give you some context, the human genome has 3.1 billion bases. So 3,100 billion bases, which means that viruses are very, very small. And so they need to really optimize what they code for because they don't have space for much more. Besides the genetic material, they have a protein capsid that basically contains the genome that they have, and that capsid can be in the form of a filamentous virus, and like we have filamentous viruses that are rod-shaped, or icosahedral viruses, or viruses in a bullet shape even. So they're very variable and they vary a lot in size, and the size of that capsid will determine the size of that genetic material. So smaller capsids have smaller genomes, and very big capsids have very big genomes. The capsids can also be surrounded by lipid envelopes and contain different other proteins that the virus will carry with them. Like I mentioned, viruses need a living cell to be able to complete their infectious cycle, so they need a host to do that. And they are very diverse in the type of hosts they choose. They can infect all forms of cellular organisms, including animals, plants, bacteria, and fungi. So why do we study viruses? First of all, viruses are everywhere. They are the most abundant biological entities in our planet. And so, like I said, they infect all cellular organisms. They are the major cause of human and plant diseases. And so the best example is what we're experiencing right now with COVID-19 epidemic. But we have also very famous viruses like poliovirus, HIV, foot and mouth disease virus, even potato virus Y, which have caused major epidemics in our food production systems. Here in this image, there's an example of a particular virus that was very famous. It is the tulip rakin virus that infects tulips and causes these patterns in the flowers. And they were very famous in like 17th century Holland because they were very expensive because of those patterns. And some people argue they were even almost responsible for a financial crisis. But the more we learn about viruses, the more we understand that they are not just parasites. Viruses can also be mutualistic. And with the development of new sequencing tools, we are uncovering a very large number of new viruses, and we're discovering that they can be commensal, so they can be in a host but not really damage the host, and they can even be symbiotic or beneficial to that host and provide some kind of benefit to that host, whether that is providing survival skills to survive, to better tolerate environmental conditions or climate conditions, such as like droughts or temperature, or even confer some tolerance to other pathogens. So you've been hearing a lot about microbiomes and the microbiome of organisms. Well, we have a virome too. Every organism has a virome, and we are just beginning to uncover like the implications of that. We also need to understand viruses in order to control them. We cannot control what we don't understand. Viruses are very, very useful biotechnological tools. Since they mimic the processes of cells, we have used viruses to basically discover many important aspects of cellular and molecular biology. And also viruses are great vectors for gene transfer and gene expression, because they transmit their genetic material to the cell. Viruses are very good tools to deliver information to cells, and that's why we use them in things like vaccine delivery, or even in the future for gene therapy. Viruses also play major roles in global ecology and the evolution of the biosphere. So in order to understand life and evolution, we need to understand viruses. And if I haven't convinced you by now that why we should study viruses, we should just study them because they're very cool. This is an electron microscope image of a bacteriophage, a virus that infects bacteria. And I honestly think it's one of the most beautiful images in biology. These are these little perfect, efficient machines that look, this is a real picture. They look like they were just made in a robotics company. So I particularly study plant viruses, and I would argue that although they're not as famous as animal viruses, they are just as important, if not more important. The plant viruses make up almost half of the plant disease causing pathogens, and they cost us around $30 billion yearly to control them. These crop losses are, of course, disproportionately affecting food insecure populations in developing countries, which causes devastating nutritional impact. And so plant epidemics are often associated with famine events. And just to give you some examples, right now we are going through a bad epidemic of cassava mosaic disease. It's disease that affects cassava, and it's caused by a complex of viruses. And cassava is a staple food in Africa. More than 500 million people depend on cassava for calorie intake. So epidemics related to cassava are very often the causes of famine events. Another important viral epidemic that we have right now is the cacao swollen shoot virus, which is affecting cacao crops in West Africa. And West Africa produces around 70% of the world production of cacao. So this is definitely something we need to pay attention to. And finally, the famous papaya ring spot virus, which almost devastated the papaya industry in Hawaii, and that led to the development of the famous GMO papaya, which is around as 90% of the papaya we consume right now, and basically saved the papaya industry in Hawaii. So symptoms of viral disease include mosaic patterns, chlorotic or necrotic lesions, yellowing stripes or streaks, leaf rolling, fruit and vegetable malformation, or stunting. So although viruses infect a very diverse number of hosts, they all share a common general infection cycle. And I'm going to walk you through the general steps that are shared by all viral infectious cycles. So first, the virus has to get to the cell and has to enter the cell. Once it enters, it has to deliver its information, and that information needs to be decoded. And that's what we call translation. So in translation, the virus has the message, and that message it's produced into proteins. Once those proteins are produced, then the virus replicates its genome and then assembles new capsids and new viral particles that are later released to infect another host. The focus of my work is in the decoding step. We study how viruses produce those proteins, synthesize those proteins, and that step is a key step in the viral life cycle, because the production of those proteins is what it will determine later the ability of the virus to, for example, defend the defense mechanisms of the host. It will provide all the enzymes that they need to replicate their genomes. So it's definitely one of the most important steps. So let me talk to you a little bit about protein translation. The dogma in molecular biology says that the flow of information goes from DNA, and then it's made into RNA, and from RNA, it's made into proteins. And so protein synthesis is probably the most important step. It's arguably the most important step in every cellular life cycle, because once proteins are synthesized, they are involved and responsible for basically all cellular processes. They are involved in basically all cellular processes, including growth, senescence, maintenance, defense, reproduction, and energy production. So this means that if we are trying to understand how organisms work, we first need to understand how proteins function and how they are made. And so basically the protein synthesis process is based on an mRNA molecule, that is this RNA, and the mRNA contains a set of instructions on how to produce that protein. So this is the coding part of the message that will be translated into a protein. And this is the untranslated region of that messenger RNA that will contain the instruction on how to initiate the protein synthesis. And so this untranslated region basically recruits a lot of cellular factors that then recruit the ribosome, which is think of it as the protein making machine of the cell. And once that ribosome is recruited, it scans through the message until it finds a signal that tells it to start protein synthesis. So this is a simplified illustration on how we call a canonical translation, and that's how most cellular proteins are produced. Viruses, on the other hand, have evolved, let's say, alternative mechanisms to that translation. Some viruses, not all of them, because they need to reduce the number of cellular translation factors they need to hijack. Think of it as, remember I told you viruses like basically depend entirely on the host and its machinery to complete their replication cycle. So the less amount of things they need to sequester, the more effective they would be. The more effective they would be, but also they will have less chances of being, for example, detected by the cell defense machinery. Viruses are also under strong selective pressure to optimize their coding capacity. That means that they are under a lot of pressure to have smaller genomes So they don't really have space in their genome to code for all the enzymes that they need in order to carry on a canonical translation process. Finally, viruses are under a lot of pressure to rapidly and massively synthesize proteins because they need to basically exponentially assemble new particles to be released. So they need to constantly and rapidly produce these proteins. And so for those reasons, viruses have basically adapted or modified or create variations of those instructions in their UTRs, and they have developed different ways to carry translation that deviate a little bit from the canonical cellular translation. And these alternative little segments that they create to deviate from the cell translation can provide them different advantages, like, for example, reallocating all the cell resources so that they favor viral translation instead of cellular translation. So they basically can shut off cell translation, and that also means that the cell has to basically turn off all the protein production. That would mean proteins produced to defend against the virus, and so that is only beneficial to the virus. It also probably gives speeds up that protein production process, and that's what the virus wants. And so that is the focus of my research. I basically try to understand what and how these little segments that we call translation enhancer elements are. And so I try to understand how they work, where can we find them, which virus has them, which mRNAs have them, how can we find them? So basically developing methods to try to understand how we can systematically screen for translation enhancer elements. And most importantly, how can we use these little elements for our benefit? So that leads me to why should we study translation? And so in terms of plants, we need to understand how they respond to a stress or the environment. And remember, I told you proteins are involved in virtually all cellular processes. So understanding how those proteins are made would probably give us alternative ways to manipulate crops in order to improve, for example, nutritional qualities, longevity, stress tolerance, and resistance to pathogens. Non-canonical mechanisms are also present in plants. So all those alternative mechanisms I just mentioned are also present in plants, but they are not as well studied. So we really need to understand when they work, how they work in plants. In terms of viral translation, we need to understand how viruses produce their proteins in order to design effective and sustainable control strategies. That being by breeding or engineer resistant plants, because if we know the requirements or the needs of a virus in a host, we can take those and design strategies to fight them. Or by designing antiviral compounds or molecules to specifically target the virus. So viruses are very hard to control because, like I told you, they basically depend and use all the cellular translation in different machineries. So if you target the virus, you're basically targeting the cell. And so if we find specific ways that only viruses use that are very good, like potential control steps for the virus. Also, viruses, these little, they're not always little, these translation enhancer elements would probably be good biotechnological tools for large-scale protein production in plant systems, because if we find a way to use them like viruses use them to massively produce proteins, then we can find good systems to, again, large-scale produce proteins, for example, vaccines. And finally, these translation mechanisms can have clues to understand the evolutionary history of viruses and organisms, as they seem to have related aspects. So recently, we have characterized a distinct translation enhancer element in Triticum mosaic virus. This is an RNA virus that infects wheat. It causes these streaks that you see here in the wheat leaves. And what we were able to find is that Triticum has in its untranslated region enhancer element that is around 30 nucleotides long, and that we think it recruits the ribosome directly by base pairing, so that is different on how the cell would do it, and that probably confers advantages to triticum, as they would allow it to cluster the ribosomes and again favor viral translation, shutting down or reducing cellular translation. We also found that this translation enhancer element that we saw in triticum is found in other animal viruses, but triticum, as you remember, is a plant virus. So this might suggest a common evolutionary history among these viruses. This translation enhancer element is probably a good candidate at this point to target it to control triticum in wheat, triticum mosaic virus in wheat. And so right now what we're doing is we're trying to screen for translation enhancer elements in different viruses, and a lot like viruses that we can find in public databases. And by finding these translation enhancer elements, our goals are to find common mechanisms between viruses that would allow us, for example, to design broad spectrum antiviral compounds or antiviral strategies that would allow us to target multiple viruses at the same time, but also to understand the diversity in protein translation strategies, because we keep learning that viruses are the perfect model to understand, again, how general translation works. So to conclude, I hope I didn't talk too fast. Protein production is a key step in viral infectious cycle. Understanding the different mechanisms virus use and their deviation from cellular translation can give us specific targets to design those control strategies. So I hope that's the take-home message that I'm leaving you guys with. But also that viruses are phenomenal protein-making machines, and we can find a way to use them in our advantage if we learn how they work. And last but not least, that plant viruses can be as devastating as animal viruses, if not more. So we have to pay as much attention to plant viruses as we do to animal viruses, because plant health is everyone's health. And so before I talk about a little bit about Colombia, I want to acknowledge my lab and my PI, Orly Rakutundra, who is the person who guides me through all this project. And so now I was asked to talk a little bit about Colombia or the culture or whatever I wanted to talk about. So I think I have to start with one of my favorite facts about Colombia, which is that Colombia is the second most biodiverse country in the world, but it has around 10% of the whole planet's biodiversity. We are ranked first in orchid production and birds, or not orchid production, orchids in general, and birds. And we're ranked second in freshwater fish, in amphibians, in butterflies, and in plants. We have all types of climates, all types of ecosystems. So if you like nature, Colombia should definitely be in your bucket list. I come from Medellin, and if you know me, you probably know that if I start talking about Medellin, I might not stop. I was telling Claudia before we started that Medellin is the city of the eternal spring. We have a temperature of around 22 Celsius, that's like 72 Fahrenheit all year round. So it's pretty much the perfect weather. And if you're in Wisconsin right now or anywhere where you have winter, you can definitely appreciate that. We live in a valley, so we are surrounded by mountains all over. And a lot of people know that Colombia is famous for its coffee, and Antioquia, which is the department where Medellin is one of the largest coffee producers in Colombia. You can see in this picture our beautiful mountain ranges. And I wanted to show you a typical dish in Colombia to talk about food in Medellin. This is the bandeja paisa. It's an enormous amount of food. It has 2,200 calories. I'm not into calorie counting or any of that. I just wanted to give you a dimension on how much food this is. And it was probably started by farmers or people working the land to just like, because they didn't have the opportunity to have more than one meal a day, but now people eat it just as one of their meals. And I don't know if I have time, but I like the idea of giving you guys a recipe. These are Panda Jukas. They are my favorite pastry of Medellin. They are super easy to make. And so I will quickly tell you what they are. They are cassava and cheese, like bread, little bites of bread. Um, that's what you would eat in Colombia in the time between lunch and dinner with like a coffee or a hot chocolate. And they're very easy to make. This is like the American friendly version, because everything you have here you can just buy it in any grocery store. You need tapioca flour and cheese, preferably mozzarella cheese or queso fresco. um, and you just mix all this in a bowl. You make little cheese balls and you bake them, and believe me, you won't regret it. They are just phenomenal. They are crunchy outside, fluffy inside, and you're going to love them. So, with that, thank you everyone for coming. I would be happy to answer any questions if you have them, and if you want to email me or contact me anyway, I'm always happy to chat about viruses, Medellin, or recipes.
Thank you very much, Elena. I'm going to stop the recording and then open the floor for any questions.